So let's talk about the internal structures that you have inside of your eukaryotic cell. First thing we're going to talk about is the cytoplasm. Okay, it's the same type of jelly or gel-like matrix, matrix being the stuff, the substance, as inside your prokaryotic cell. It is considered as lying between the cytoplasmic membrane and the nuclear membrane. So remember, the nucleus is internal, it's isolated, it has its own kind of space. This is the GUI between that cytoplasmic membrane on the outside and where that nucleus is on the inside. Okay, so here's a cell, right? And it's a eukaryotic cell, and I can tell that because it's got a nucleus and it's got organelles. Okay, so the cytoplasm is pretty much, so here's my nucleus in purple in the middle. Everything between the membrane of this purple and the membrane of the cell. So all of this GUI and everything floating inside of it is going to be my cytoplasm. If I was just talking about the GUI by itself, just the blue, okay, just the blue without any of this stuff in here, okay, then you would call it the cytosol. There is a difference between the two, just so you're aware. Now, the nucleus. In this case, this lavender purple thing is going to be the nucleus. They are only found in eukaryotic cells. Okay, remember that because eukaryo means true nucleus, right? Prokaryo is before the nucleus. So you will only find them in eukaryotic cells, and they are separated from the cytoplasm by what we call the nuclear membrane. It is an envelope that surrounds the nucleus. So if you look here, if this were solid, you can see the purple by itself. It's basically encasing everything that's in here, and it's separating, giving it its own space from what's in the rest of the cell in the cytoplasm. Okay, it contains two primary structures. Okay, two primary important parts. One is the chromatin, which is the DNA. It's suspended in its own GUI that we call the nucleoplasm because it's inside the nucleus. Okay. And then you have the nucleolus, which is a dense body that floats inside of that nucleus. Now, the nucleolus is where we make ribosomal RNA. Okay. It is an important part, structure that you find inside of ribosomes. Okay. So if you look here, see this dense this dense thing right here, this is the nucleolus. This is where we make our ribosomal RNA. Whereas all of the light blue squiggles are going to be the genetic material or the DNA for this specific cell. Both of these things are isolated in the nucleus as their own space or their own room. The DNA itself is floating inside of jelly. It's not like it's air that is inside of this. So it's got its own nucleoplasm, GUI, that helps to kind of suspend the DNA and helps to suspend the nucleolus. All right. So the next structures we're going to talk about are the endoplasmic reticulum and the Golgi apparatus. Okay. The endoplasmic reticulum is a series of membranes used in protein production, transport, and storage. So, protein production, making proteins, protein transport, moving it from point A to point B, and protein storage. We're going to put it away until we need it later. We have two types of endoplasmic reticulum, rough and smooth. Rough endoplasmic reticulum originates from the outer membrane of the nuclear envelope. So it literally is continuous with the nuclear envelope. The reason that we call it the rough endoplasmic reticulum is because it's speckled with ribosomes. Ribosomes actually sit on top of this membrane, um, and the ribosomes are responsible for the protein synthesis that is happening. Now, once they are made, then the endoplasmic reticulum can kind of package them and transport them where they need to go. Um, Either they can go to the endoplasmic reticulum as storage, or they can go to the cell membrane, or they can go to another part of the cell. But the rough endoplasmic reticulum is kind of like the post office that packages and then sends to wherever it needs to go. Now, the smooth endoplasmic reticulum is actually the same set of membranes, except it's called smooth because there are no ribosomes on it, and its function is completely different. So we have no ribosomes, that's the reason that it's smooth, and it's used in nutrient processing. It can be used to do the chemical reactions that we need to process nutrients, and it can be used to make lipids and to store lipids if we need it. So in this cell, okay, we have, the rough endoplasmic reticulum. So you can see that it is membranes that are stacked on each other, kind of like pancakes. But you can also see that it is covered in ribosomes. So usually, at least everything that I've seen, the rough endoplasmic reticulum is always right next door to our nucleus. Okay? So this is where we are going to make proteins, package them, and then ship them to wherever they need to go. Now, up above that, here, okay, see how there's no ribosomes on it? This would be the smooth endoplasmic reticulum. Notice that they are technically the same color, that same dark, deep navy blue. So you know they are technically the same thing, but one has ribosomes, one doesn't. So the one that has ribosomes is going to be rough. The one that doesn't is going to be smooth. Now, the Golgi apparatus. This is the site of protein modification and shipping. So remember I said when we make the protein, we ship it somewhere. Normally, when the rough endoplasmic reticulum makes its protein, it's not finalized yet. It's not finished. You have to remember that a protein is going to be a complex machine that does something. It's going to be an enzyme that breaks something down. It's going to be an enzyme that takes different parts and puts them together. It's going to be a transport protein that moves things in and out of the cell. It is a complex thing. It's not just, I'm going to put yarn together and go poof, and it's going to be whatever it's going to be. It's going to be a wadded up pile of yarn. It actually has to be put together in the right way. Some parts have to be cut out. Some things have to actually be inserted to get it to work correctly. All of that work happens in the Golgi. Okay, that's why we say modification and then shipping. So you've got a series of membranes. It's similar in shape to the endoplasmic reticulum, and it actually works in close conjunction with the endoplasmic reticulum. The endoplasmic reticulum sends out transitional vesicles containing proteins and lipids, which dock with the Golgi. They actually meet up with the Golgi. The proteins and lipids leave the vesicles and enter the Golgi for modification by the addition of polysaccharides, sugars, or and lipids. So I might add bits of lipid to them. Once completed with modification, the proteins and the lipids are pinched off into condensing vesicles for the use inside the cell, or they're transported via transport vesicle to the membrane of the cell or completely outside. So again, if we're looking at this, here's my endoplasmic reticulum, right? Rough, smooth. See these little blue bubbles right here? There we go. They're actually transporting up to this guy. Again, it's a bunch of membranes stacked on each other. Again, notice the colors the same. This is where these guys dock. And as it goes through these layers, it's getting modified. It might get parts taken out. Like I said, it might have things put in, the polysaccharides or the lipids, like I said, until it's finished and ready to use. Now, once it's finished, it gets put into another vesicle. This vesicle may go inside the cell to get used in some way. Maybe I need enzymes for breaking down polysaccharides, or it can actually get sent up here to the membrane where it'll fuse and maybe make something like a transport protein that allows things into and out of the cell. Finalization of that protein that we made here is going to happen here. Okay, so in the rough endoplasmic reticulum, made a long chain of proteins. You've got a bundle of yarn. In the Golgi, I'm going to put that yarn together in the right way so that it functions the way I want it to function. So it works the way I want it to work. Okay, the mitochondria. Mitochondria, when you're in grade school, you learn are the powerhouse of the cell. So what does that mean? There's a difference between memorizing something to regurgitate it and understanding what something means. So why do we call it the powerhouse of the cell? It's responsible for generating ATP, okay? Adenosine triphosphate. So ATP is like gasoline. We put gasoline in our car to get our car to go, right? ATP is the chemical that we use to energize activities inside of our cell. Okay. This is one of those things that I was talking about where I said, or this is what I was talking about last lecture when I said, making ATP, I can give you 25 cents, I can give you 50 cents, or I can give you 36 to 38 quarters. This is the best way to do that. So it's an awesome organelle to have. Okay. It is unique. This is one of those that I said kind of gives credence to the idea that, or I should say, credibility to the idea that more complex cells engulfed less complex cells, and those less complex cells ended up being beneficial, so we kept them. Okay. And again, you can see that internal membrane and then this outer membrane. They are unique in the fact that they can divide independently. The cell doesn't have to tell them you divide now. They can do it whenever they feel like doing it. 2, they have their own DNA. Remember I told you, another thing that lends credibility to the idea of this engulfing endosymbiosis is the fact that these things have their own genetics, okay? And then finally, the ribosomes that you see inside of mitochondria are the 70S, which is the size you see inside of bacteria. In eukaryotic cells, the ribosomes are bigger, okay? So if you look here, here, this is a mitochondria. You can see the squiggly inside and you can see this membrane out here. That squiggly inside is its own independent membrane. And then the outside here is a second membrane. So it's a double membrane structure. Okay. Mitochondria can be different shapes. In most models, you see them kind of looking like pinto beans. But in reality, they can scrunch up and become a circle. They can elongate and look like worms. It's not necessarily a static structure that only has the one shape. But the way that you will always know it's a mitochondria is because of that squiggly inside. That's the internal membrane. Sorry. Okay. Note the interesting structure of the mitochondria, the double membrane, an inner and an outer membrane, and an outer membrane and a folded inner membrane. The folds in the inner membrane are called cristae, okay? Cristae. And they're the sites where ATP is made via the process of cellular respiration. Cellular respiration uses oxygen, okay? So cellular respiration has to have that in order to work. It's like saying, I've got a car, I don't have any spark plugs in it, it works fine. No, it doesn't. I have to have oxygen to get the mitochondria to work. You have to have spark plugs to get your car to work. All right. Now, other internal structures. We've got chloroplasts. Chloroplasts look like this, okay? And they are found in photosynthetic eukaryotic organisms. Photo means light, synthetic, synthesize, or to make, okay? So they convert light energy into chemical energy for the cell to use. In other words, I can be a plant sitting out in the sun, and as light comes into me, I use water, carbon dioxide, and that light to make sugar. Okay? It's one of the reasons why we eat plants, because they make food for us. Well, technically, they're making food for themselves, but we take advantage of the fact that they do that. For single-celled eukaryotic organisms that have chloroplasts in them, they can do the same thing. They can use light so that they make their own food. Okay? So that's one. Hang on one second. Sorry, I wanted to remember. Well, I remembered I had a plant cell. So if you look, you can see these guys right here, these green structures. Chloroplasts are often depicted as being green because they absorb the light that isn't green. So they are absorbing red light, purple light, blue light. The green is the only one they don't absorb, so our eyes perceive them as being green. Are chloroplasts exclusively green? No, because we've got things like red algae that actually have a red or brown algae that have a brown color to them, in which case what they're absorbing might be slightly different, but they're still photosynthetic, meaning that they are still using light to create chemical energy. Okay? I'm sorry, guys. So, ribosomes. These are the protein factories. These are the guys that read the instruction manual and put the parts together in the right order. So, what are they reading? They're reading messenger RNA. Okay. Larger size, a larger size is found in eukaryotic organisms versus prokaryotic. So remember, prokaryotic are at 70S. In eukaryotic, you have 80S. They're larger. You kind of have two types. You have some that kind of float free in the GUI of the cell in the cytoplasm, but you also have some that are attached to the endoplasmic reticulum. So you've got free and you've got bound ribosomes. And as we already discussed, mitochondria have their own ribosomes that are different than what you find in the cell. So when you look in the cell, you've got some bound to my endoplasmic reticulum. But if you look down here, see these peachy colored things? And up here, where are you? You've got these peachy colored things. Even over here, these are going to be your free floating ribosomes. Now, what's the difference? Normally, the ones that are attached to the endoplasmic reticulum are things that are either going to be put into the membrane, so they're going to kind of work outward, or they're going to be like excreted out of the cell. The ones that are free floating inside the cytoplasm are going to be the ones that make the proteins stay inside my cell. Okay? So, the cytoskeleton. Cytoskeleton is a complex network of microtubules and filaments that help with cell structure, cell movement, organelle placement, intracellular, inside cellular, the cell, movement of molecules and vesicles. They can be composed of actin myofilaments. Actin myofilaments are very, very small. They kind of look like two pearl necklaces that are wrapped around each other. They're very tiny. Microtubules. Microtubules are a bit more complex. It kind of looks like Twizzlers, except solid. There's no hole in the middle. Okay? You've got something with a little bit more girth to it. And intermediate filaments. You know what? I'm wrong. Intermediate filaments are the solid Twizzlers. Microtubules are the pipes. Okay? Each type differs in shape and function that it performs. So, cell structure, like our skeleton gives us our structure, cytoskeleton helps the cell to maintain its shape. So, for example, we have within us different cell shapes and types. So, some of them kind of resemble a palm tree, where you've got a whole bunch of cell at the top, a long extension at the bottom, and then something that kind of looks like roots. It has to stay that shape. So we've got these cytoskeletal elements that help us to maintain that. Now, earlier I said cellular movement. Some cells can actually move by themselves by kind of oozing. If you want to see that, look up amoeboid movement on YouTube. I'll see if I can post that with this. But they basically are able to shift their shape around so that they can surround things. Kind of like what we saw at the very beginning when we engulfed the prokaryotic cell. Okay? Something else. It can be used to move internal structures from one point inside the cell to another point inside the cell, almost like train tracks or a highway system. The organelle may kind of attach to it and then use it to get to another location, let go, and then just stay in that place. So we're not just talking about gross movement, the whole cell moving. Sometimes we're talking about things inside the cell moving from one point to another. All right.